Welcome to Biocanic's Health Intelligence Exchange community, where leading practitioners in integrative health approaches will discuss how they solve the most challenging and common health issues. These regular Q&A discussions are designed to take a deep dive into case studies and integrative approaches in order to elevate the collective knowledge of both experienced and new practitioners alike. Biocanic's Health Intelligence Exchange, your community for functional medicine.
All right, welcome to the Health Intelligence Exchange, everyone. I'm Jeremy Malek of Biocanic, and today I'm joined by our guest, Dana Walsh, Senior Microbiome Scientist for NirvanaBiome. We'll be exploring how whole genome shotgun sequencing transforms microbiome testing. Be sure to subscribe wherever you're watching or listening so you never miss an episode. And for those of you joining us live today, we'll move into a Q&A session towards the end of this conversation. So feel free to post those comments wherever you're watching. So drop commenting those comments. NirvanaBiome uses whole genome shotgun sequencing, also called WGS, for consistent high-quality results in genome identification. They're able to characterize the microbes found in your gut and understand some of the functions they may be performing. They find estimates of the amount of the microbes that were identified in your sample and can compare your microbes to a group of more than 800 healthy people. All right, thank you so much, Dana. Let's go ahead and jump right in. Well, actually, just quickly, do you want to just introduce your background, how you got into being a microbiome scientist?
Sure. So starting in undergrad, I was interested in microbiology. So that kind of started me on a path thinking I was going to go into microbiology. Ended up in the microbiome. I had not heard of it pre-graduate school, and it's fascinating to learn about all the microbes that are present in the human body and the roles that they play in your health and everywhere on the body, whether it's the gut, the vagina, your skin. So graduate school, I was at University of North Carolina, Chapel Hill. I did airway microbiome. And I've always done a mixture until my current position. I've done a mixture of wet bench, so tissue culture, mouse models, molecular biology, in combination with bioinformatics and statistical analysis. So airway microbiome there, did a postdoc at Mayo Clinic, and that was ovarian cancer and endometrial cancer microbiomes using human studies as well as mouse models, tissue culture models. And finally, I worked for a pharmaceutical company for a couple of years and then landed with NirvanaBiome, so doing statistical analysis for them. And also if you buy a gut test or a vaginal test through us, that comes with a free consult. So you'll schedule that with me, and I will sit down with you one-on-one and walk you through your report, help you interpret it, make sense of it, help you figure out like what your action steps might be, what the changes in your gut microbiome mean. And then, from there, what your next steps are, whether that's further testing or supplements, whatever we decide on.
Awesome. Okay, so let's kind of set the stage. So, I think the gut microbiome, gut health, gut-brain axis is all the rage these days. So, we talked a little, in the intro, we talked about whole genome shotgun sequencing, or WGS. So maybe why don't we kind of start with the foundation of what is whole genome shotgun sequencing and how does it differ from the other microbiome testing methods?
Mm-hmm. Yeah. So whole genome shotgun sequencing means we're sequencing all the available genetic material in a sample. So prior to WGS becoming more affordable, the main method was amplicon sequencing. So in that case, we were specifically targeting a bacterial gene. So then it's the 16S gene specifically. So what that is based on is that gene has conserved and also hypervariable regions. So you have primers that will, attach to those conserved regions. And then the idea is you can identify what bacteria there are there from the hypervariable regions. But that, suffers from primer bias, as well as really you can get down to genus level with good confidence. Whereas whole genome sequencing, because we're sequencing more of the genome, you can get to species level and even strain level with a lot higher confidence. In addition, you're sequencing everything. So you're sequencing any genes, functional genes that might be there. So we can not only identify bacterial species, but also look at like what functional roles might they be playing. This is DNA level, so we don't know what proteins or metabolites they're making, but yes, it allows us to predict.
Awesome. And maybe for those of us that don't remember biology class, do you just want to walk through genus, just walk through the hierarchy of all different things and what those mean?
Definitely, yes. Yeah, forget people don't work with this every day. Yes.
Yeah.
So my mnemonic is kings play chess on fine green sand. I'm sure you have something like that you learned as an undergrad, but yes, phylum, class, order, species, genus, family, genus, species. Yes. So we can look at all those taxonomic levels. And with species, we can even get down to strain level with whole genome sequencing.
Awesome. And then when we talk about kind of, from your perspective, what does whole genome sequencing capture that clinicians are often missing with traditional stool tests?
So I believe your traditional stool tests are probably PCR based. So you're targeting a specific gene. So they're very specific. And especially like that can be totally fine if you know what you're looking for and you want to detect a specific organism. But if you don't know and you just kind of want to get a feel for like what bacteria are present in this sample, whole genome sequencing is great because in theory, we are sequencing everything that's there and we can identify everything that's there and look at functionally what roles they might be playing.
Mm-hmm. Okay. And then why, so, kind of talking through the whole hierarchy of the different bacteria, why does strain level resolution matter clinically and not just species level identification?
Different strains might, they might have different functionalities, or maybe they have different resistance genes or different virulence factors. So like, you might be able to identify a certain species and say, okay, this bug is here, but is it the bug that produces or can potentially produce this specific toxin or is resistant to this specific drug? So strain level, you can actually identify that. Another advantage is that if you are making a probiotic or some type of supplement or something that has a bacteria in it, and you want to be able to track specifically your strain of interest, we can identify that strain and track it and make sure, like, did it actually get into the gut microbiome, or did it get to where you wanted it to go? And is it staying in the gut over time if it's a probiotic? Or, actually the advantage is actually being able to identify that, yes, this one is there.
Awesome. And then if we talk a little bit deeper about whole genome shotgun sequencing, how does it improve accuracy with identifying pathogens, commensal, opportunistic organisms in stool samples?
Well, I think this relates to the fact that we can differentiate species and even strain level pretty accurately, because that'll help you identify more specifically what bug you're looking at. The other thing too is when we're looking on the functional side, and actually like looking at, okay, this particular species has these predicted functions. These look more like what like a commensal species might produce, or these look more like what a virulent species might produce. So that's another way it can be advantageous.
Okay. And then maybe in a second, we'll kind of jump over to kind of going through the report itself. But, what role does WGS play in detecting those microbial imbalances that might be overlooked with symptom-based or market-limited testing?
I think the advantage, so really, I think the methods are complementary because you still want your PCR specific testing, right, to identify for sure, like this is the bug that's there, and maybe you want to quantify it. Like that whole genome sequencing is relative abundance. There are ways in which we can get at absolute, but for the most part, it's relative. I just forgot where I was going with that. Can you repeat the question?
So what is it like? How having whole genome shotgun sequencing data? How does that really play in kind of detecting those imbalances versus just symptom-based or market-limited testing?
Yes. So the fact is that you can look at like the community as a whole. So rather than just, okay, this specific species is changing, that maybe it's related to the disease, but now with whole genome sequencing, you can look at the balance of microbes that are there, like the whole community, like what is shifting because this particular microbe is present that you're detecting with your traditional stool testing. Is it shifting the gut microbiome. And then, if you treat that, can you tell that it's shifting back to a healthier state? So it kind of helps you assess the overall health of the gut microbiome.
Awesome. And if you want to kind of pull up the report that we want to go through, and then so we're, kind of get the practitioner view and how to review kind of that whole genome sequencing and how to kind of go through that clinical insight data.
Yes, I can do that. So let me find, here's the report. Okay, so I've got it up. Are you guys able to see it yet?
Yep, there we go.
Okay, cool. So this is a NirvanaBiome report. Do you want me to walk kind of through this whole thing, or I guess I can just go and, feel free to stop me. So with this one that we're looking at is actually from someone who she had some type of like food poisoning. She had some type of bug, which you can definitely see. So this is her sample on the right. This is our healthy control group on the left. So our healthy control group, healthy. I say healthy, they're more accurately disease-free. So there are about 800 mostly Caucasian people living in the US, so Western-based diet. But this is what we typically see at the phylum level. So this is our highest level of classification. And I like this because it gives you kind of a broad overview of, are there big changes going on here in comparison to our control set? So generally we see Bacteroidetes and Firmicutes in this kind of 1-to-1 ratio. And then these other less abundant, less dominant phyla. So Actinobacteria, Proteobacteria, Verrucomicrobia. So this includes Akkermansia. You might be familiar with that one. And then, of course, your Proteobacteria include your gram-negatives, Klebsiella, E. coli, things of that nature. So this person's sample on the right, you can see like she definitely does not have that 1-to-1 ratio. She's got, Firmicutes is pretty relatively normal, 58%, but she's got a reduction in Bacteroidetes here, and then an expansion in the Proteobacteria. So like if I hadn't known ahead of time that, she had an infection, I'd be like, oh boy, something is not right here because we don't expect this much Proteobacteria. So that's a red flag to me immediately. The second half of this report, I don't have to go through this in extreme detail, but we do list like the phyla by name. This is the interquartile range of our control group, so you can kind of see where your sample falls. So here's the relative abundance of your sample. And then we actually give you a percentile here. So yeah, you can see that Firmicutes is quite high. Actinobacteria, Proteobacteria, they're all quite high. As we continue through, we also look at family level, so getting more specific. I'm not going to go through this in much detail for the sake of time. But we also have genus level. So here you might be seeing bacteria that names you recognize. We don't show this for species level just because these charts become unreadable, but we do provide a full list of the species we detect and their abundances. So here you can see the patient or the subject, however you want to refer to her. So lots of Citrobacter. That's unusual. I don't usually see that high dominance of Citrobacter in a healthy stool sample. At least there's a little bit higher Prevotella, but Prevotella is a normal commensal gut species. We're seeing Bifido. We're seeing this Lachnospiraceae. And then in our control group, we have Bacteroides, Phocaeocola, Allostypes. So these are all your normal gut commensals. So this Citrobacter here and kind of the expansion of Prevotella stand out to me.
So when you're looking at it, you're kind of trying to visually match the pattern.
Yes. Yeah. So kind of just doing a comparison and thinking about what stands out here as there's a lot of inter-individual variability. So what you'll see is that, rarely is your healthy gut sample, for example, going to match our control group because this is like the mean of 800 individuals.
Right.
But what I am doing is I'm kind of looking for our major players. And I'm also looking to see, is there anything else here that stands out to me as not typical for the healthy gut? So Bifido, Bacteroides, Phocaeocola, Blaudia, these are all normal. It's the Citrobacter that stands out to me here.
And then maybe from a kind of a practical application perspective, if practitioners been using, a 16S stool microbiome test and now they move to this, are there common misconceptions or things that they would trip over moving to whole genome, or is that transition relatively straightforward, kind of an apples to apples?
Yes, it's pretty straightforward. Like as far as the taxonomic results go, you're gonna, look at that and kind of interpret that in a similar way. The big difference is whole genome does tend to be a little bit more accurate, and you get more specificity getting, down to strain level. But the big advantage is the addition of the functional content that you don't get with the 16S sequencing. And we do have that in this report. What I can do is, if we want to go through the functional a little bit, I can skip down to that. But we also have in here our gut microbiome index. How much time do I have to go through?
Plenty of time. Take your time.
Okay, great. So this is that list of individual species that we detected. And you can see like Citrobacter is pretty high. Even this Prevotella copri is pretty high at 6%. So most of the time at the individual species level, it's 3% and lower is what I see in the healthy gut. So when I'm looking at this list, again, I'm looking for things like this. Like, is there something odd that dominates the community? So the Citrobacter, the Prevotella copri. But then I'll just scan through the list to see, are there other bacteria that stand out to me as not normal to see in a healthy gut? So it does take some familiarity with the species that should be there, which again is why we offer the one-on-one consults, so I can sit down and help you with this. Yes, but really it's these Citrobacter species and the Prevotella that are standing out to me in this particular sample.
And then is there like thresholds or like if something's above a certain, or are you just looking for distribution, relative contribution?
It's kind of relative contribution. I would say there's not like a hard threshold. Like we do give these interquartile ranges of the control group, so you can kind of see like what generally should you expect in a healthy person. But like if someone exceeds this range, that's not necessarily cause for alarm, because again, there's a lot of variability from person to person. Yes, but the way I kind of interpret the report is I am kind of making notes as I go through of what I should be paying attention to. And then when we get to the functional section, we'll relate some of these changes we saw at the taxa level into the functional profiling.
Awesome.
All right. So this next section is our gut microbiome index. So this is an index that we developed at NirvanaBiome. We've actually made this is an older report. We've made this out of 100 now. You see it's out of 30 here. So it's out of 100 now. It makes a little bit more sense that way. But any score above 20 is considered excellent. So even though this person does have clearly has, some imbalances here, they still get this score. And that's because we're looking at these, what I think of as broad community level metrics. So here's alpha and beta diversity. These are taken from ecology. These are used a lot in microbiome science as just broad. I think of them as broad descriptors of the microbiome. So alpha diversity, this is going to be like number of unique samples. So you see we actually enumerate this for you in the report. There was 132 in this person's sample. And this is what our control group looks like. So interquartile range, median here. So, they're actually still within this control group. So they still get they get like a mid score for alpha diversity. Generally with gut samples, the higher your diversity, the healthier your sample. Beta diversity, this is a comparison. So what we're looking at now is the abundance and then the prevalence, or like different species in the sample of your entire community, and how similar is that to other samples. So this cloud in gray is our healthy control group, and you can see like there is quite a spread here. Like, so again, I was talking about variation between person to person, and you can clearly see that here. But really with the subject sample, we want to see this fall within the cloud. So the score will be lower if it's like off to the edges or floating outside the cloud. So this person actually gets a high score. And the final piece is our organisms of interest index. So in our current report, we do list all of the organisms that we're actually looking for, and you'll see like a lot of these are species that are associated with infections. So Klebsiella will be there, Escherichia coli. We also look for protease, fungi, and I think there's some viruses in that list as well. But I can tell you that generally C. diff, Bacteroides fragilis, other C. diff species, obviously, like those are also commensal, but they tend to be picked up. So they are detected in our healthy group. This Citrobacter looks like it's also in our healthy group. Like it's at relatively low abundance in this person. So there was that other Citrobacter species, if you remember in our species section, yes, that had higher abundance than this one. But essentially, like this person gets a higher score because they have fewer organisms of interest, and also they fall pretty much within the range of our control group.
Why are those the organisms of interest?
It's a list that we've kind of hand curated where we know they might be associated with disease or infection. So, and yeah, this is why we say this is not diagnostic, but like, you know, we're detecting C. diff DNA in your stool sample, for example.
So, and I think you might have touched on this, but so in that case, C. diff is slightly above the relative abundance. Like,
yeah,
how would that kind of impact on the organism interest index? Like, what is indicative of a problem? How would you kind of think about that?
I kind of base, like, this is all calculated into this overall score. So, you know, the overall score is great. Like, this is an 8. The higher, the better. And really, like, I talked about variation between people. So, like, this 0.17 really isn't that far above our control range here. And everything else, I'm just looking at it real quick. Yeah, everything else falls within the range. So, like, for the most part, all of these species we're also detecting in our control group. So they're more likely than not commensal species that we're picking up and aren't indicative of a problem. Clearly, we were not looking for the Citrobacter that maybe caused her gut issues, but yes. And I disclaimer that I knew that she had an actual clinical test to detect that. So yes. All right, so I can move into the functional section next, unless any other questions before I move on.
No, go ahead.
Okay. So the functional section, like, we can actually do more than what's presented here, but we kind of just chose some important functional pathways to look at in this report. So we have a section of short-chain fatty acids as well as B vitamins that your gut bugs produce. And you'll have like an overall score for both of those. And then we break those down as well by the individual vitamins and fatty acids. So this is the score for your sample, your patient sample in this report. And then this is our control group range here. So this particular person actually gets a very high score for overall short-chain fatty acid production. And then we can go down and actually break that down into the individual short-chain fatty acids. So butyrate for this person was also quite high. Propionate, however, it was kind of average. So, like, you know, falling into this average range is fine. Even falling into the low range is fine, because it's not necessarily a one-to-one relationship of, like, this is DNA level. Like, we don't know how the DNA we're detecting in these bacterial communities translate to what's actually getting produced, you know, at the protein or metabolite level. So, you know, if we're detecting it, you have bugs that can potentially make it. Acetate is also quite high for this individual. And then lactate is the final one, which is also average. So, you know, overall, short-chain fatty acid production looks fine in this person.
And so what would be the kind of presentation or implications of being low or not detected?
In that case, that's where I might say, okay, well, we should look back. So I'll get to this in a second. We also have a list of keystone species where we know they have specific roles, like making these short-chain fatty acids or B vitamins, things of that nature. So I might, kind of connect those and say, okay, you have low butyrate, for example, that we're detecting in your functional profiling. And then if we're not seeing many species that can produce butyrate in this person's sample, I might go, well, maybe think about a probiotic with specific butyrate producing species, or taking a butyrate supplement, for example. So this is where, kind of connecting those helps me make recommendations for, like, is there a supplement that might help you, like, improve these levels?
Okay.
I'll just keep scrolling.
Yeah.
Yep. So this person also has high polyamine production. We also look at GABA that the gut microbiome can produce. Average for this person, so that's important for your gut-brain axis. So, glutathione, antioxidants also high. Vitamin synthesis. So this is that kind of high-level summary range of B vitamin production. It's high in this person, and then we break these down by individual B vitamins. So B1 average, B2 average. B5 is low in this person. So, B vitamins, obviously, you could take a B vitamin, a B complex, if you were concerned about that. B6 is average. B7 is high. B9 is average. B12 is average. Yeah. So overall, functionally, this person seems fine with the exception of that expansion of Citrobacter. And then this is that keystone species of interest section I mentioned. So this is where we pull specific bacteria that we know have these roles. They've been shown robustly and consistently in the literature. So obviously, Akkermansia muciniphila, you're familiar. I'm sure a lot of you are familiar with its role in metabolism. This person actually did not have Akkermansia muciniphila. So I do recall that we had the Verrucomicrobia phyla, and I believe that's only Akkermansia. So there are a couple other Akkermansia species. So this person probably has Akkermansia based on that, just not muciniphila. And that's important to note, because just because this says not detected, that doesn't necessarily mean your gut microbiome is missing this specific function, because microbes have redundant functional roles. So you might have other microbes that can play those roles.
So in this case, would somebody then go on to like a pendulum for Akkermansia supplementation, or? If
they were interested in that, sure. I mean, in my opinion, it doesn't hurt to try a probiotic.
Okay.
And then you can, the other thing you can do is use this test to see where's your gut microbiome at, what might you want to target to supplement, whether that's probiotics or specific supplements, and then come back to us, I don't know, 3 months later after you've been taking it, and see, did that actually change things and make a difference and improve your gut microbiome. I'm just going to scroll through, see what else. So here are our short chain fatty acid producers, and you can see this person, she's got this butyrate producer, but not this butyrate producer. So, clearly showing functional redundancy here. Otherwise, these two are both cellulose degraders, not detected in her gut. However, I would have to go back to the specific species list, because what I'm looking at here is these are both Ruminococcus species. And if she had other Ruminococci, she probably still has this function. Yep. And the last page is just a summary of the numbers, really. So percentages of the organisms of interest, your diversity scores, keystone species, and then the functions.
And then what about pathogens, H. pylori? What about any of those other markers?
We would pick those up. So that goes back to our organisms of interest index. So I know H. pylori is in that list. And I know we didn't have, I'll just scroll back up. We don't have the full list. Actually, let me stop sharing for a second, because I have a newer report I can show you that does have that full list. Whoops. Let's see if I can do this. Share screen. It was on. Okay. All right. I'm sharing the screen. Are you guys able to see this report?
Yep. Yeah, we can see it now.
So this is a newer one. It's organized slightly differently. We have a few notes here just on some specifics in the report. Oh, is this really small though? Maybe I need to zoom it in. And you can see here now our gut microbiome index is out of 100. So it's total to 100. But I was going to scroll down to our organisms of interest. Okay, so here it is. So now you can see there's quite an extensive list of species that we're actually looking for. I believe H. pylori is in here, right here. Yep. So we are looking for it. E. coli, C. diff, Campylobacter, Salmonella, Vibrio. So yes, all of these that you might be associated with disease. Proteus, Candida species, looking at fungi. So yes, the caveat here, obviously this is not diagnostic, but also we're looking at the DNA levels. So we don't know if these organisms are alive or dead when we're detecting them.
Mm-hmm. And so what does that, like, how would somebody try to interpret that? Like, what would they be looking for, kind of the clinical implication?
If you detect it, you mean?
Yeah, I mean, so if it's alive or dead, like what is the impact and how you kind of think about it?
I guess what I would say is, like, we're detecting DNA here. We don't know if it's alive or dead. So like if it's a high percentage and you're concerned about it, then I would say you should probably actually get a targeted clinical test to see, like, is this something of concern, especially if there were symptoms associated.
Okay. Awesome. So what are some of the kind of, now that people, get into this, what are some common misconceptions practitioners would have when they first start working with a whole genome sequence like this?
I do get a lot of people who are very concerned about, the ranges and thinking, oh, it has to be within that range or it's bad. So it takes a little time to wrap your head around the fact that, like, interpreting microbiome results is a little, I don't know the right word for it, but it's not black and white, for example. It's you kind of have to take the community as a whole into account because it's a relative comparison. So that can be a little bit difficult to wrap your head around, I think, as a clinician starting to look into this.
Awesome. And then just kind of thinking about what role does whole genome sequencing play in detecting those imbalances that might, how do you, let's say somebody is suffering from, gas, pain, and bloating after eating. How would you run this and you're looking for diversification? How would you think about, like, what would be the approach for kind of how you would go through that process?
Like, you're saying if they have, like, gas or bloating as a symptom and they want to take a look at their gut microbiome and kind of figure out, like, what might be the issue?
Yeah, exactly.
Yes. So first of all, like, when I go through these reports, it is very helpful for me to know, like, what are your concerns before I sit down with you, because then I can look into the literature a little bit and say, like, are there specific species that we know that are associated with these symptoms? I have looked into gas and bloating before, and off the top of my head, I can't remember what those species are, but, like, then I'm kind of keeping an eye out for, are we seeing an increase in an abundance of these specific species that, might be problematic? And then what other changes, like, are there functional changes that we're seeing? Like, do you have low functional ranges for, I don't know, specific short-chain fatty acids, and we need to supplement that back, or we need to add a specific fiber into the diet that would support the beneficial bugs that would then push out these species associated with the symptoms?
Mm-hmm. Yeah, I mean, because I'm kind of thinking, like, in most applications, people are coming in for the first time. They're largely, if they're going, sometimes GI map is a core component of the individual methodology, but a lot of times it's like, hey, loose stool, gas, pain, and bloating, burping all the time, other things. And so I'm trying to think of how does that fit into, the clinical application when you get that first test, maybe not the trend, but that initial test. So kind of that methodology for what a practitioner would go through in kind of thinking about it.
I mean, if I was sitting down with a first NirvanaBiome test for someone with those symptoms, I would essentially just be looking for what are the changes I see in comparison to our control group, and then noting, like, are these species that are changing a lot? Is there something that's dominating the community? And then taking that, maybe back into the literature to see, do we know that this species is associated with these symptoms, and is there something we can do to reduce that? Usually with diet or supplement or probiotic.
And then so obviously once you have 2 tests or more, you're going to have that trend to be able to kind of look at what is changing, which is going to be a lot more insightful. How long do you need to wait? How much is transient variability? How much is sustained transformation? And like, what does that time interval look like?
So that is kind of a tricky question. It kind of depends on your question, what you're interested in, what you want to see. So we know like the gut microbiome can change from like even between meals. So another thing is like try to take it, at the same time of day to have that consistency. If it's like a lifestyle change, like a diet change, or you're taking a probiotic, I don't know. I guess I'd give it like 3 months, and we could definitely see if something has kind of established or is kind of hanging out there, or has shifted, other aspects of your community, like other species have gone up or down, and are those beneficial or not?
And one of the kind of the hot topics in the functional personalized health space is really around women's health.
Mm-hmm.
So how does the estrobolome, like in women's health, how does that relate to the testing? Perimenopause, PCOS, how would you kind of see differences within those tests?
Like as far as they relate to the gut microbiome and the gut test specifically?
Yeah. Would you see differences based upon those particular conditions or phases of life?
Potentially. I do believe menopause does change the gut microbiome. I don't get a lot of post-menopause samples, so it's not something I've looked into in a lot of depth. But it's also not something that like we've incorporated into the report as of yet. Like we do have a vaginal test, so obviously it might play a bigger role there, but I think we would potentially see an impact of those treatments in our gut testing, but it would be something where I would need to dig into the background a little bit more, and what bugs are we expecting this to have an effect on.
And then kind of like you talked a little bit about before we started, you work with a lot of naturopaths. What are some of kind of like the unique cases that you've seen, just kind of the highlights that really stuck out between different individual cases?
Well, I can tell you a lot of people come to us with IBS in general, with constipation symptoms, bloating. One that has stood out. I've had a couple of kids, actually. Those kind of stand out because we don't get a lot of kids. Some people, like, they're interested in, can we look at, like, are there potential changes associated with like autism symptoms, and like are there signs we're seeing there? We do have an autism test as well, so it's not something I do a lot, but yes, that's one of the interesting ones.
What makes that one different from the general test?
That's where, so it's very similar, but we're actually identifying specific organisms that have been associated with autism and how they're changing, and like are their abundances increased or decreased in association with autism. So I guess it's a more targeted report.
Okay. And then, how do you think whole genome sequencing help guides those clinical approaches in terms of the protocol versus just like a protocol-based intervention? For example, somebody's got SIBO, there's like a typical protocol versus how do you see this kind of changing the landscape in terms of guiding the protocol?
Yeah, well, clearly it's not diagnostic or right at this point. But I mean, I could definitely see it like looking at the community as a whole, and especially with conditions where we know there's a gut associated component, IBS, C. diff infection, like it might give you insights into what specific species are changing. Are they increased or decreased? Are they causing these symptoms? Do we need to target them specifically? Rather than like, you have these symptoms and you're just going to target with this specific drug. Microbiome sequencing might allow you to see, is there a specific microbe you should be targeting?
And then in parallel, how does whole genome sequencing fit within the other clinical data points like labs, symptoms, or lifestyle factors? Like, how would you kind of think about synthesizing this into clinical practice for our providers alongside of the other blood
Yeah.
and everything else?
Yep. So we have physicians and naturopaths obviously who do this now, but I think they use it more as not diagnostic, more like informative, like how is your general gut health doing? Are there things that are shifting that we need to be concerned about? Do we need to be making lifestyle changes to shift that gut microbiome back to a healthier state? Consider taking a probiotic. So I feel like it's more of a whole health and wellness maybe indicator rather than guiding specific diagnostics.
Okay. How do you kind of, what are you guys excited about going forward in terms of advancement in the microbiome space? What are you seeing coming on the horizon? What do you guys kind of think about implementing or going forward with?
Yeah. I think there's a whole lot more we could be doing with our functional section, and adding in different functional components and different functional groups to expand on that and make it more informative. The other cool thing is metabolomics. Adding metabolomics in would be super cool because then you'd get what are the metabolites that the gut microbiome is producing and how are those changing in response to your lifestyle changes or a drug or whatever you're interested in. So eventually, having that kind of tandem testing with whole genome and metabolomics and integrating that into the report, I think could be super informative for like a whole health systems level kind of analysis.
Okay. Awesome. Any new bacteria that people are finding? Like, is there, as kind of this is becoming more into the mainstream from a research perspective, are they finding new bacteria? Or have we found all the bacteria? Now we're just trying to understand what it does.
Well, there are lots of unnamed species in our database. So, I mean, I think, yes, to some extent we're finding new species and, sequencing can identify all the species, right? And we might not have those in our database necessarily, but, then you got to actually do the biochemical, the microbiology testing to classify that species. So it's a slow process. So there's a lot right now that just stay as like a species and a number, and you might see that in the report, and it just means it's an unclassified species.
Okay. So there is new insights to be found.
Yeah.
I feel like it's kind of like the ocean. Like,
yeah, it is.
Been studying the ocean for years, and then all of a sudden they find a new animal that they didn't know existed.
Yes. And it's like it's hard for me to pinpoint like a specific one because like I see all these, I don't know if they're necessarily new or we just haven't given them a name yet, but like, species name and then the numbers like indicating that we don't know what this species is, but it's a bacterial species and
of unknown origin.
Yes. Yeah.
Well, awesome. Hey, this has been a great discussion. So I want to make sure we get to our questions. So thanks to everybody who's been posting questions. We'll get to as many as we can. And if you're watching this as a replay, feel free to post questions wherever you're watching. So let's get to our first question. For practitioners considering adding whole genome sequencing for stool testing into their practice, what would you say is the biggest shift in mindset that's required?
I touched on this, but not thinking about like the numbers and the ranges in the report as like black and white, like it has to be in this range just because the microbiome is so variable from person to person. So like I said, like if a person is a little below that range or a little above that range, but overall their microbiome looks pretty normal, then it's probably normal for them. The other thing too is like when you're doing repeat testing, you can tell like, okay, this is consistently high for this individual, but they're not experiencing symptoms. They're generally healthy. So then, this person just has more of that particular species. So I think that's a big thing.
And actually going back to one of the things that you said when you were going through the test report, you said your normals is like 800 without disease.
Yes. Yeah.
Largely Caucasian in the US. What would be the implications of having that broader data set or non-Caucasian living in South Africa? What would that look like?
So actually what would be really cool is if we could tailor our control population to like your patient's samples. So I don't know, if you're Native American and you're 20 years old, like we'd have a decently sized control set to compare them to, or specifically if you're in other parts of the world where your diet is probably quite different, it would be important to have that comparison group. So that's where I think like it'd be nice to have those tailored control groups at some point in the future. But it's difficult because you got to do the research to get that data as well. So.
Mm-hmm. And I think you mentioned that it's non-disease, but it's controlled. But then that's the point of, I think is it the beta diversity plot where it's kind of like a scatter plot?
Yeah. The reason I say it's non-disease and not necessarily healthy is because right. Like the definition of the healthy microbiome is kind of contested, right? Like what is a healthy microbiome? Because there's so much variability from person to person.
Yeah, no, I've said that quite a bit, which is what is that? Who knows what healthy is, right? So, yep. And we're still relatively early in the science in terms of trying to understand what that means. Mm-hmm, mm-hmm. So I think that is a great point. And that's kind of just why I wanted to say that. But another question, can someone use this test alongside another stool test or other functional test to gain some clarity? I think you might have touched on this a little bit.
Yeah.
Doing that.
Yeah, definitely. I actually think it's super advantageous to do it in tandem with other tests. So whether that's your targeted PCR-based tests where you can actually quantify your species of interest, and then look at that gut microbiome tests and see, is it off balance because we have a high quantity of whatever species you're looking at, or the metabolomics where you're looking at, I don't know, bile acid or short chain fatty acid panel, and then you're looking at the functional report and saying, does it make sense that, I don't know, we have low butyrate in the context of knowing what this person is actually producing.
Awesome. Yeah, it'd be interesting to run. I think it was 5 or 6 years ago, I actually ran 3 stool tests off the same sample. And it was interesting. It was 80% overlap. And then there was one that was using, quote, at the time, AI interpretation, and they had conflicting interpretations of my B vitamin levels.
Oh, interesting.
Yeah.
Did you submit them all to the same company or different?
So one was a traditional 16S. The other 2 were partner. They were the same testing laboratory that you're obviously familiar with from the lab testing, but they were 2 different interpretation reports of this from the same lab.
Oh, okay. Interesting. Yeah. I mean, I would definitely expect like Whole-Genome and 16S to have some differences. Like 80% overlap is great. Yeah. Just because of the differences in the methodology. Like even from lab to lab, there can be differences just because databases and algorithms are different. So.
Yeah. And then actually, this is another question that came in. That's a great question, is how should practitioners think about microbial microbial diversity in context? Is higher or is better? And what this is always drives me nuts. And I think this comes to the point of it's still early. What does normal mean? Mm-hmm. The concept that's in the kind of the conversation around gut health is the more bacteria, the better. Feed your gut, healthy gut, constantly feed your gut with fiber. Like, how would you kind of think about that in context of diversity?
Yeah, so generally, a more diverse gut microbiome is associated with better health outcomes as of this point in time. I don't know that anyone has like discovered there's some kind of a threshold to like, is there a too high diversity in the gut? Obviously, other areas of the body, it's different. So like vaginal, for example, you actually want low diversity there. So you want lactobacilli species to dominate. Skin is somewhere in between, but higher diversity is a little bit better there. Higher oral diversity is better. So yeah, depends on the body site for sure.
Yeah, and are you familiar with I think it was a study out of Stanford that came out like 2 or 3 years ago where they, quote unquote, ate the rainbow, and then they also ate fermented foods. And I can't remember, it's the Sonnenberg lab.
Oh, you know what? I think I heard of it. It's been a while. I'd have to go back and read it again. But. And
I think it was feeding it with just fibers, fruits, and vegetables and fruits. I had no impact on diversity. It was only at eating fermented foods that actually increased diversity.
Yeah, and I would bet it's because you're providing both, the bacteria plus the food they're eating. So you're boosting those good species.
Yeah. Next question, can whole genome sequencing data help us distinguish between transient dysbiosis and entrenched ecosystem imbalance?
That's a good question. That's where I would say that your like longitudinal testing over time would be super useful, because then you'd get a feel for, I don't know, did something disrupt this person's gut microbiome short term? Because I would expect it would go back to more of a normal state in a, I don't know, shortish period of time. How long is, I guess, up for interpretation. But then if it was more of an established dysbiosis, yes, like it would stick around for like years maybe and be consistent. So yes, I think it could definitely show you that.
Yeah. It'll be interesting to see, as now that more of the technology is more cost effective, and it'd be great for you to kind of talk about that. But to see what the overall impact on all of this kind of prebiotic, probiotic, postbiotic, which biotic type of thing, and how much of an impact it actually has from an actual residence biome versus just transient impact.
Exactly. So from my data analysis experience, I can tell you most probiotics, especially if they're single strain, are transient. So while you're taking it, we can detect it there. We know it's there. We can pick it up. But if you stop taking it, it just gets washed out, because if you have a healthy gut flora, it's diverse already, and all those bacteria are occupying their niches, and there's not really a niche for this probiotic to establish itself. I think other studies have shown that if you have more strains in your probiotic, more diversity in the probiotic itself, it's more likely that you might get species engrafting. But yeah, like unless you continue to take that probiotic, at least with a healthy gut, a gut that has something established there. Like if you've taken an antibiotic and wiped out your gut diversity, then like a couple different strains might be able to establish, because now you've opened those niches, for other bugs to establish themselves.
Yeah. And there's so much there's so much debate related to how much, I've got X thousands of different of this strain or that strain versus live probiotic versus the kind of the probiotic you buy at Costco in a gigantic bottle for $10.
Yes, exactly. Right? Yeah. I am definitely a proponent of probiotics with more strains in them. So like, I think I actually take one now that has at least 12, if not more, different species in it. So, but obviously like that fiber component, the prebiotic component, like actually supporting the bugs and their functions and what they're doing. I think that should happen in tandem, because it helps support those healthy bugs.
Yeah. And then I think another question here, how can whole genome sequencing insights support metabolic, immune, or neuroinflammation cases differently than traditional stool testing?
Well, we can pick up some of those markers if the gut bugs produce them. So like that's another thing, like with the functional side of things, where we could potentially look at like immunomodulatory functions that we're finding in the microbiome. Obviously, we're not quantifying it like a lab test would. So it would be more of a comparative thing where we're looking at, are we seeing these in higher abundances relative to the entire group?
Awesome. Well, listen, this has been a great discussion. I want to thank everybody who joined us today and those who asked questions. If you're still watching as a replay, you can still post your questions. We'll make sure to get them over. Thank you again, Dana. We really appreciate you joining. So where can people find out more about you, NirvanaBiome, and how to kind of get going?
We do have a website, NirvanaBiome.com. You can also email us at hello@NirvanaBiome.com. I am on LinkedIn as Dana Walsh. I guess I can share like my email if that's of interest later. But
yeah. Well, we'll definitely get that posted in the comments, and practitioners sign up at the website, get more information. Is that correct?
Yeah, yes. And definitely feel free to reach out by email if you have specific questions to me or to NirvanaBiome. So
awesome. Well, this has been great. Thanks everybody for joining us. Really appreciate it. Thank you again, Dana. And
thanks for having me.
catch us in 2 weeks with our next guest on our Hicks live stream. And we're really excited to see you then. Awesome. Thanks, everybody.